just introduce me.
Okay. Yeah.
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 Awesome. Oh.
Just one quick stuff.
Yes.
On the email,
Yes.
I think it will be nice. So, I effort to figure out one.
Was it literally 3 to 1?
This is GL3 actually.
Ah, okay. Thanks for that.
No
We'll,
worries. Essentially doesn't exist. So we were like kind of trying to figure out the puzzle. So I hope no one else goes off.
Thank you indeed. Thanks very much indeed.
No worries.
Excellent. Right, thank you very much indeed for coming to the 4th coding club of the series. Today is because it's slightly electric subject matter. First of all, we have Hoon from mathematics. We'll be talking about pipelining. She will explain a lot more in her talk. And then we have James, who will be talking about some system workflow tools that will help us enable pipelining work as well. So hand over to Yin. Thank you. Finish at about 20 past.
Okay.
You find questions.
Okay. Good afternoon. I'm Hoon. I'm currently a postdoc at the School of Mathematics. I joined the university September last year, and before that, I was working in New York at Mount Sinai School of Medicine. The main project of my work in New York was to build whole cell models, and in order to do that, we actually had to build a software pipeline, and which is the work that I'll be sharing with you today. As you can imagine, building a pipeline wasn't a solo effort. So I'd like to start by acknowledging all the people who were involved. So these are the people who contributed substantially to some of the software packages that I'll be sharing with you today. So this is kind of the outline of my presentation today. I'll briefly describe the research problem that we are trying to solve. Then we'll go on to discussing some of the pipeline architecture. I'll also briefly share the computational performance of this pipeline, and I'll finish up with some of the challenges and lessons that I've learned in this work. So as I mentioned earlier, my project was actually to build whole cell models. So what is whole cell model? Our definition of a whole cell model is a computational model of single cells that mechanistically represents all the genes and gene functions in the cell. And when I say mechanistic representation, I was referring to all the biochemical reactions and biophysical interactions in the cell. So why do we want to build such models? So this model is inspired by engineering practice, where it's very common to use model simulation to help us design products and services. So the idea here is, if we have a model where we include all the genes and gene functions, then perhaps we could use such models to help us design microbes with special traits that we're interested in. For example, maybe we want the microbes to be able to produce some drugs or vaccines that would be useful for human consumptions. From a medical perspective, our vision is that now that genome sequencing is getting cheaper, we envision that perhaps at some point in the future, it would be possible to say a patient comes in, we sequence their genome and then put that information into a model, run the model and try to design the most effective therapies that is personalized to that patient. So that is our vision. But what is the current state of the art? So currently there are only a few whole cell models out there. The first one was published in 2012 by a group at Stanford University, and that was for bacteria with about 400 genes. And over the years, these are the other whole cell models that have been published so far. What I didn't show in this table is that this model, for example, it took a few PhD students, and in the US, a PhD usually takes about 5 to 6 years. A few PhD students, 5 to 6 years, manually curating the data and manually coding all the codes for the model. And that's for a bacteria with 400 genes. Imagine if you had to build something for human cells with 20,000 genes, how long is it going to take if we have to do this manually? And so with this realization, that was why we decided that we need something to help us automate or semi-automate the whole model building process. And so this is the pipeline that we have built. Again, we were inspired by practices in software engineering. So we started from specifications of what we would like this pipeline to do. We wanted this pipeline to be scalable so that we can use the same pipeline to build both a small bacteria model as well as a large human cell model. We also wanted this to be modular so that individual pipeline can be reused and repurposed. And there are also other things that we felt was important. So one of the problems in research, especially in scientific research and modeling, is that many of those results are not reproducible. There are papers publishing models out there. If you look at those equations and try to reproduce them, sometimes you can't reproduce the figures of the simulations. And some of those are because the data that they use, it's not really clear where it comes from. What are the assumptions made by the modelers during the model building process? And so in this pipeline, we wanted to have something where we can include all the metadata, including the sources of the data. So this is what we currently have. So the first step, we created some software packages that allow us to combine quantitative and qualitative data into a knowledge base. And then this data in the knowledge base is used to automatically generate submodels. And the quantitative data is used to populate initial conditions of the state variables. And finally, we combine this into a whole cell model that can be used for simulation. In the next few slides, I'll go into more details on each of these steps. So the first step was to build a knowledge base. As you can imagine, all the data that we need to build a model is scattered in all sorts of public databases and also papers. So in this step, we needed methods to help us obtain those data. And we also wanted a single schema for representing all this diverse data. And more importantly, as I mentioned, we wanted a pipeline where we can trace the sources of all the data. In this case, we wanted a structured knowledge base that is easy for us to navigate. And this is what we have done. So we have developed something, an API called Datanator, that allows us to query data from public databases that store information about cells. We have also developed a schema called WCKB, which is a knowledge-based schema. I'll provide more detail about this schema later. And the result of this knowledge base comes in two formats. The first format is a set of Python objects, which can be written into an Excel file that's really more convenient for storage and visual inspection. But this Excel file can also be read back into Python objects in memory so that we can do further analysis or model development. As I said earlier, many of those data are not in public databases. There are many data that is still scattered in, say, a TSV file, CSV file, and Excel spreadsheets as supplementary information in those papers. And in this case, our pipeline, it would not be easy to have a pipeline with software that automatically just get those data. So for this, the modeler, in this case myself, I had to write custom codes to parse those data manually. So I mentioned earlier about the WCKB schema. So WCKB, it was built upon another software packages that we developed called object tables. This use object relational mapping. The way it works is that we have different classes of information. For example, we have a class for chromosomes, and these are the attributes for the properties of members of these classes. And we know that each chromosome contains many genes. So the relationship is that a member of a chromosome class has a one-to-many relationship to a member of a gene. And a member of gene can have many transcripts, whereas each transcript can only produce one protein, therefore the relationship is 1-to-1. So with this kind of a relational mapping, it made it easier for us to traverse across this relationship if you want to, say, trace information about a particular protein and trace it back to a chromosome and a gene. So that's what it looks like in the schema in Python objects. So here I show you like a snapshot of a screenshot of what it looks like in Excel. So each class has its own is a worksheet, and each row is a member of an instance of that class. and each column is an attribute of that class. So that's how we develop a knowledge base. The next step is to then use the knowledge base to help us automatically generate the submodels. And in this case, again, we need more methods to help us do that. So the way we decided what is a submodel is based on categorizing individual subcellular processes as one submodel. For example, transcribing a gene is one submodel, translating into a protein is another submodel. And each of these, it has different reaction patterns, which I'll mention. I'll describe next what I meant by reaction patterns. And in order to do that, so I have developed a Python packages called WC Model Gen. These packages consist of various modules. Each module is used to generate one submodel. This model gen use the knowledge base as an input, and then generate the submodel, which is represented as another schema called WCLang. And again, WCLang comes in two formats, Python objects and Excel spreadsheet. So I'll just give you a brief explanation of what is the steps required in generating each submodel. So basically it has 3 steps. The first step is to generate the reactions. So I mentioned earlier about reaction patterns. So here I show you an example of a protein degradation reaction. So to those of you who may not be familiar with biology, so in the cells we have proteins, which is basically a polymer made up of many amino acids in different sequence. Now each protein has its own length and different sequence of the amino acids. If you were to degrade the protein, what happens is that all the bonds between the amino acids will be chopped down, and that requires a water molecule. Now, depending on the length and sequence of that protein, it will require a slightly different numbers of water molecules, and the product of the amino acids will also be different. And because we have genome annotation data in the knowledge base, it was quite straightforward to just use those information to help us automatically determine the length, the sequence, and therefore this is the number of, say, water molecules for this particular protein that is required to degrade this protein. So that was how we generate the reactions. The next step was then to generate the rate law or rate equations. I wouldn't go into details other than to say that we try to incorporate as much information as possible. For example, the enzymes that is required to catalyze the reaction, we use that and generate a rate law to incorporate that into the rate equations. And finally, the last step was to calibrate the submodel to the quantitative data in the knowledge base. So that is what the pipeline looks like. So how well does it work? So I've used this pipeline to build a wholesale model of human embryonic stem cells. I currently have 2 versions of the model. The first model was a full model, which contains all the 20,000 genes. And so this table here shows you, for example, in this full model, the number of reactions is more than 200,000. And it took me more than 1 week on an HPC to generate that. And that is from the knowledge base. So I didn't run it like at one go. I generate the knowledge base, store it in an Excel file, and then read it back into Python object and then generate the model. And that took me more than a week. And because of that, I decided to also build a smaller version of the model. In this case, because we have a pipeline, it was quite straightforward to write some simple script to say, okay, I only want these genes to be included and these reactions to be included, and then rerun the pipeline. And I was able to build a smaller models with only about 1,600 genes. And this one contains about 31,000 reactions, and it took me about 2 days. So I'm happy to talk a bit more about.
As on the coding club thing.
Okay, sorry. Yes. So this is what this current performance of the pipeline. We actually didn't do a lot of performance profiling, so I believe there might be possibility of improving the performance still. I'm happy to talk a bit more on the research and the model part on another occasion afterwards, but for today, I'll just focus on the pipeline. So these are the tools and pipelines that platforms that we use for building this pipeline. So all the packages that I mentioned earlier, they are all on GitHub. Many of them are actually open source if any of you are interested. We also use Circle CI for continuous integration so that every time anybody push any changes, it will automatically run the test. And if there's any test that's failing, then we will receive an email notification. We also build all our, we also have a Docker image for our pipelines. So I guess maybe we'll hear a bit more about the tools and platforms in the next presentation. So what are the challenges of building a pipeline? I think some of these are probably not just for pipeline, but also relevant for any software development projects. Managing dependencies on third-party packages. As you can see, we have a lot of software packages, which depends on a lot of other software packages out there that are not built by us. And sometimes when they make some changes, we have to make changes on our own packages too, so that it's compatible. But besides depending on other packages, even within our own pipeline, there's a lot of dependencies, and sometimes it can be quite difficult. So throughout this project, we actually had to do a lot of like merging and splitting modules into separate packages or merging packages based on what we think is practical. And I suppose it is a matter of optimizing robustness, agility, and flexibility. And I'm not a software engineer by practice, so I believe there are probably practices out there about how to optimize this, but we actually just do it by being practical, so it's not very optimal at the moment. Another challenge is collaborative work. I think working with other people can both be very rewarding, but also challenging sometimes, which I believe some of you have experiences. So I'll just end my presentation by a few best practice and lessons that I've learned. This is actually probably just my grievances. So best practice, run your unit tests locally and ensure that the test pass before you push to GitHub. And ideally, especially if you're working in a team where you know who is developing something that depends on your packages, if possible, perhaps also test the downstream dependencies before pushing. And if not, then as a polite courtesy, maybe you can inform your colleagues that you would be pushing something that might affect their project. And if all of these are also not possible, then maybe it would be kind to not push before you go home, especially if your colleagues are going to leave slightly later than you. I've had to experience many occasions where somebody pushed something. I was about to leave home, and then the last email I received was a notification that my packages has failed because of something that somebody else did, which is not the most fun. Finally, if you're working on a branch, try to merge frequently. Or if you don't merge, then at least try to update the packages that you are using. So I have a colleague who liked to create his own branch and then he didn't normally update his Docker container, which means that his branch is not very up to date, whereas everyone else on the master branch was already up to date. And then when he merged, he had tendency to force override some of the changes that we updated. So it was quite frustrating sometimes to then have to spend a lot of time. Well, yeah, I guess many of you understand this. So yeah, it's frustrating to spend so much time just to fix tests, especially things that are not because of your own work. So anyway, thank you for your patience in listening to my grievances. This will save me a lot of therapy sessions. So with this, I thank you for your attention, and I'm happy to take any questions. Thank you.
Any questions? Thank you very much. Wait, let me just bring this closer to you.
Hi, thank you for your talk. It was really nice to listen about your work, and yeah, I share some of your grievances too, which is fun. I had a question about your reduced versus full model. Did you see much difference between the two, like when you ran applications on them? Could you benchmark, your difference between your reduced model and your full model that you computed? Do you know how they performed between each other? If there were many differences between the reduced and the full model?
What kind of performance are you referring to? Like
I don't know how you would benchmark it, or like see like functionally, like if you wanted to use these models.
Oh, okay. Well, unfortunately, I'm sorry. Hi, yeah, so in terms of performance, like computational performance, I showed the number of hours of generating the models, but I believe that's not what you meant.
Not computationally, just
Yeah.
physical.
Yes, so that's a very good question. And I have to say that unfortunately, we have not been able to actually run like real simulations for both models at the moment. As you can imagine, computationally is really, really costly. And so in that respect, from my perspective, I'm interested in research. So from my perspective, benchmarking would be on the simulation results. And because at the moment, I have not managed to do that. So
Be interesting to know.
yeah, thank you.
James?
Hi, thank you for that. That was a really good talk. Thank you. I was going to say, when you're doing anything like this, I mean, I like the approach you're taking, which is a very, formal, very solid approach. But you've got two parts, really. Your data collection, where you try and bring all your data that you're going to need in, and then your very hard maths, try to solve a lot of differential equations, I presume. But for the second part, to validate it or to at least compare it, could you substitute in some kind of machine learning model to take your inputs to try and match your outputs to at least have two paths and actually build on the work you've done in the first stage? I mean, so the question is have you looked into that?
Yes. Thank you for that question. In fact, I left this project September last year, and now the project is kind of sitting around. I don't know. I don't think anybody's working on it anymore. But as I was working on it, I realized that some of those submodels would probably improve if I could train it as a machine learning or neural network type of hybrid models. So that is actually an idea that I'm currently thinking about. And in fact, I just been rewarded a Turing Enrichment Award to organize a workshop. I am planning to organize a workshop on hybrid modeling, where we have submodels of some parts will be mechanistic, whereas some parts where we have data but not enough mechanistic understanding that could be trained using machine learning. But that is just at the moment conceptually an idea, but I haven't tried it practically. But that's a really good. Funding, I suppose. Yes. Yeah, I would be interested to chat with people who are interested in that kind of work. Yeah.
Can hook you up with James to talk that through. Any other questions online? Rory.
Thanks. This is great. I can relate to a great deal of what you've said here. I was wondering, so a fair amount of this seems to involve the kind of infrastructure work of, collating data, scraping things, making sure that they'll, work properly when they, you generate your new models. And I was wondering if you have any thoughts or, good or bad experiences on how you support that activity. So how do you make sure there are actually people available to do it without them not being able to work on things that would actually advance their careers as scientists? Because that is a problem that we run into in some of our projects.
Yeah.
A lot of exercise for this.
That's a really good question, because I suppose this is kind of like the direction that we are heading towards, like research software engineers and then researchers, and now even many startup and companies providing services. I haven't really thought much about that part of the problem. Yeah, I would really like to be involved in being part of the community in just going towards the same idea of making it work for everybody and open source. I really like open source. I think that's where we should head towards. But yeah, more practical thoughts, I have not thought of it. But I'll be happy to chat more in details, or if other people have any ideas, I would be happy to be involved. Yeah, thank you.
Thank you again.
Thank you.
For our next speaker, we've got James.
in. Okay, so the other way around. I believe so, yeah.
We have liftoff.
Oh, got it. Sorry, it's being recorded. Oh, I think I'll need the mic as well, won't I?
There you go.
Thank you. Okay, hopefully you can hear that. Oh. Is that still working? Can people hear me? I haven't muted it because it's gone under my lapel. Good. Okay, excellent. Right. Welcome to the second part of this. So this is very much a kind of a practical introduction to just a couple of tools that I use extensively on HPC systems, specifically BlueBEAR. I kept changing HPC to BlueBEAR and back again, and I settled on HPC because I thought this is a generic tool. So I'm talking about, actually, just a quick introduction. My name is James Carpenter. I am one of the research application specialists in the research software group, which is part of Advanced Research Computing at University. Okay. So I'm talking about two tools which people may or may not be familiar with. First is TMUX, the terminal multiplexer. And the second is not VS Code generally, but specifically remote development in VS Code, which I use quite a lot. And I will introduce it from a kind of native VS Code perspective, and then I will present a few caveats to running it natively, particularly on HPC systems, and then look at a kind of a BlueBEAR centric system, which is running code server on Bear Portal. So why do I like TMUX? So I've been using terminal multiplexers for about 10 years since I first got my Raspberry Pi 1B. And I realized quite quickly that if I wanted to run apt-get update on that, it was going to take a very long time, and I didn't want to sit there and have to ensure that my session remained active for the duration of the apt-get. So how could I ensure that I could background it? So I Googled around and I found this thing called Screen, and I was like, this is marvelous. This is exactly what I want to do. And I worked with Screen for quite a long time, and I used a wrapper around Screen, the name of which I've completely forgotten, apart from that it begins with a B. Somebody else might know. But then when I moved to the RSG, the Research Software Group, TMUX was the kind of the tool of choice there. So I switched to using TMUX, and I haven't looked back. To paraphrase, so a terminal multiplexer essentially allows you to run multiple login sessions down essentially a single connection to the server. But it also allows you to persist connections. So I was talking about apt-get. I wanted to be able to set that running, disconnect from the server, and for it to continue. But the kind of the breadth of what you can do obviously extends beyond that. So it's used for running, in the case of on BlueBEAR, if you're running an interactive session. So if you're connecting directly to a compute node, you can do that within a TMUX on the login node. You can run a Vim session at the same time, and you can be watching, for example, HStop, and you can do this all simultaneously. And what I'm going to do now is go through a quick demo. I should quickly caveat this. So you will be running TMUX on one of the login servers. The login servers on BlueBEAR specifically are resource limited. So whilst you can run lots of tools, you can run umpteen HTOP instances, you may eventually get slightly grumpy automated emails from the system, which then tell you to stop using so much resource on a login node. So I will caveat that again when I start talking about VS Code as well, but a useful thing to bear in mind. So a quick demo of TMUX for those who are not familiar with it. So hopefully you can all see. Is that large enough, that font? Okay. I can get away with that being a little bit bigger. Okay, so I'm going to connect to BlueBEAR first. Oops, so I can spell it. I've stripped out all of my SSH keys, so it should actually force me to use my password. Good. I'm going to shrink that down a little bit now. So to initialize a TMUX session, I type tmux, and it drops me. So I can now run, for example, I mentioned htop, so I will do that. Shrink that screen down again a bit. And then if I wish to keep that process running, obviously htop isn't particularly interesting, but if you're doing something kind of more informative and you're running kind of compute that could be running in the background, I can detach from that session. So that is now running in a tmux session on the login node. To detach, you have to remember the crucial escape key for tmux, which is Control-B, which prepends pretty much every command that you want to execute in tmux, and then hit D to then detach from that. It tells me that it's detached. And then I can disconnect from my server and then reconnect. Now, I noted that that was running on BB login 02. So this is a common mistake that is made. Obviously, if you're connecting to BlueBEAR, you may hit one of 3 login nodes, so 1, 2, 3. If you set up your tmux on login 02 and then you connect to BlueBEAR again and you hit login 03, your tmux session will not be on that node. So you need to essentially remember which one you like to use. I tend to go for login 02 or 03 in general on the basis that it's the least populated in my experience. There are some caveats around that, mainly that if we have to shift login nodes around or if we have to temporarily disable one for some reason, then you may need to sort of jump to a different node. So a lesson which I have learned in the past is that don't assume that your tmux will always be there. So don't entirely rely on that session being configured and active and always kind of reconnectable to. So that's just something to bear in mind. But we'll assume that login 02 is still up from 30 seconds ago. Ooh. Where am I looking? That final hyphen. Right. Useful to remember. So this is the problem. I store all of this stuff normally in my SSH config file, so I never actually type it in. And I've probably typed in my password incorrectly. Oh, no, there we go. Okay, so I can check to see if I have any running tmux sessions by running tmux ls. And I have one that I've still got running from earlier, so I'm going to kill that so that it doesn't confuse me. A useful command that I was going to introduce later. You can kill a session with this command. So tmux kill session /t to target it, and then the name of the session, which in this case is 0. So that's the one that I started at 10:37 this morning. I should now just have one session remaining, which is there, and then I can run tmux detach, and I'm back where I left off. So that, in a nutshell, attaching and detaching from tmux sessions. Noting the host name if you do need to reconnect, because using BlueBEAR could put you somewhere that you don't want to be. So that's kind of the basic kind of principles. Where it actually starts to become useful is the ability to then split that screen up into different partitions. So you can do that. Again, the all-important escape key, Control-B, followed by the % key, will then split vertically. So that gives me 2 panes. And then I can move between them by using Control-B and the left and the right arrows. And I don't know if you can see on Zoom or even in this room, but there is a faint green and gray line down the middle. When it's green at the top, it means you're in the left, and it's green at the bottom, it means you're in the right pane. But you can also tell if you have a flashy cursor like I do in my terminal client. And then I can also split vertically. So horizontally, even that's a vertical split. I can split horizontally. If I go over to the right-hand pane, go Control-B, and the quotation mark character, the double quotation marks will then split this way. And then if I want to move these screens around, there are various different ways to do this. They don't all apply to all types of terminal client. So if you go to my favorite website, tmuxcheatsheet.com, that gives you various kind of like lists of the commands that you can use, but they don't all apply, I found, in a Mac environment. So if I want to shift, for example, the left-hand pane, I'm going to head over to the left, and then Control-B, and then I enter the command mode down in the bottom left-hand corner by hitting the colon key, and then I type resize pane, which will tab complete, but it won't offer tab completion options, I found. So if I start typing RE, if there are more than one option, it won't give me any anything that I can then tab through to. So, but once I've completed enough characters, it will give me the name of that command. And then I'm going to move this to the right by 10 cells. And I can do it like that. So that's obviously extremely clunky. My suggestion would therefore be to turn on mouse mode in Tmux, which makes it far, far easier. It also means that I can click around into the different panes that are running in a given window. So Control B, set that command again into command mode, type in set mouse on. And I can now use, hopefully you can see my cursor. I can move the mouse to travel around the different panes. And I can also, if I position the mouse in the right place, then resize the panes with the mouse, which is far, far simpler. The only slightly annoying thing is that obviously within any other kind of graphical application, you would expect to have some visual feedback when you move on the division between different panes so that you can see where you're in the right place. That doesn't happen with this, so you just need to guesstimate it, but it tends to work fairly accurately. So that's creating multiple panes and moving around them. And then I can create multiple windows within my Tmux session. Yes.
Sorry, just a quick question about mouse mode. Do you need exporting or anything like that to be on?
You, not as far as I'm aware. No. That should work natively. It doesn't. Again, it will depend. Sorry. Yes. So for those who didn't hear that, if it depends very much on your terminal client and whether it can support kind of the mouse forwarding, but most modern terminal clients should do that nowadays. So I can Control B and hit C to create a new window, and then as you can see in the bottom left-hand corner, that second window appears here. So I can run my favorite tool htop down there, and then because I'm in mouse mode, I can click on the various different windows in the session to move between them. So it's flexible, it persists. If you've got a complex setup where you've got multiple files that you want to edit, you want to see lots of content, you want to tailor an output file, it's a very good scenario for doing that. We use Tmux extensively to do all of our building on BlueBEAR. And it's a good tool. So I'm going to detach from this and I'm going to throw in a little bonus for any Mac users in the room. So I am running a terminal client called iTerm2, and iTerm2 has native Tmux support, and I can additionally pass the -CC option, which puts it into control mode, and then instead of whereas before I had to click around and I had the windows listed at the bottom, the windows are now turned into tabs, and the actual panes are a kind of a Mac native panes, and I can drag around, and I actually do in this case get the boundary. So there are slight advantages there, and also if you're running multiple sessions on the Mac client, you can navigate between those with a GUI as well. So it's another option. So that is Tmux in a very quick nutshell. For those who don't use it, I would suggest that it's a very useful tool to be aware of. For those who do, hopefully you've learned something a little bit new. And if you're a Mac user, hopefully you like the sound of iTerm2. It's a very good client, although a little bit feature rich in my opinion. Yes. Can I see it over here? Give me a second. Oh, I was going to put it over there. Would it be frowned upon to copy our own doc files for Tmux Vim configuration onto BlueBEAR? No, not at all. No, I mean, however you find it easiest to do your work. I don't know where to look because I'm talking to someone out there. But no, not at all. So the only file that we discourage editing of is .bashrc, or if you are doing that, be very, very careful with what you put into it. But certainly loading configs with Vim and Tmux is absolutely fine. I don't know if my colleagues want to add anything to that. Okay.
Because
Yes, so that was basically, for those who didn't hear, be very careful about version compatibility. You may find that some features, particularly because we're running CentOS, and CentOS is quite a conservative operating system, it tends to run relatively sort of, certainly not bleeding edge versions of software applications. So just make sure that you've not included some feature on your home system that's then incompatible with BlueBEAR. But certainly maintaining your own sort of set of tools is a good idea. And in my environment, I have all of my dot files, dot .vims and dot .tmoxes in a Git repo, and then I symlink from my home directory to those, so that I can version control them as well, which is always to be advised. Right. If anyone doesn't, if nobody has any more questions regarding Tmox, I'm going to move on to Visual Studio. So I imagine that most people here are probably are familiar with Visual Studio Code. I was a great Atom user for a long time, and then I discovered Visual Studio Code, having resisted it for quite a long time, because as a Mac user, I tend to resist Microsoft stuff in general. But actually, it's a very, very good piece of software. Microsoft is great in some ways for this type of thing. Perfect. It has a large development community. It's a highly extended piece of software. I think the reason why I ended up going towards it in the end, because all of the Atom plugins that I was using, the developers were saying, we build this on Atom, but actually just go and run it on VS Code because it runs better. And that pushed me from one to the other. So we're talking specifically this time about running remotes in VS Code. So this is the ability to run your local version of VS Code and connect via SSH to a remote server. So in the case of editing files on BlueBEAR, your options are opening a terminal client. Actually, there are several options, but I'm going to very quickly cover them. Opening a terminal client and using something such as Vim, which in the majority of cases is great, but you might want a more feature-rich IDE. If you are a BEAR portal user, we have a file sort of navigator built into BEAR portal, which also includes 2 levels actually of development environment. But again, it's learning another development environment. If you're already familiar with VS Code, sticking with what you know can be useful. The other option being you run VS Code locally on your system, and then you mount using Samba your remote your project directory onto your local computer, and then work off that. But again, I prefer not to use Samba where I can avoid it, and with other HPC systems that might not be an option. So Baskerville, for example, doesn't have a Samba server, or doesn't have Samba access to its storage. So if you want to connect to Baskerville, using a kind of an SSH native method is wise. So first thing you need to do is ensure that you have the correct extension. So you essentially filter your extension list by remote, and then remote SSH is the one that you need, and it gives you a very useful little video on how to configure it. But essentially, you will then get a remote explorer. You can either go this way, or I tend to use the bottom left button because I can find it a bit easier, particularly when this list of in the left-hand tabs increases. By default, it will pull all of the hosts that you have configured in your .ssh/config file, which tends to sit in home directory .ssh. I have disabled that, so I've overridden it for this because I've got about 20 or 30 different servers configured in there, and it makes this list very long and difficult to manage. So I've put in a clean config file for my SSH, so I can create a connection from scratch. So again, I'm going to SSH my username. And if you don't have a .ssh/config file, this will create one for you and insert the correct lines to make this connection. I will also forward my agent. So as you can see, it's created this file for me. So I've got it in my temp SSH config. But essentially, if you're not familiar with SSH config files, this is broadly what they look like. You can do lots of very powerful things with them. The man page is very useful for that. So I now have an SSH target, which is BlueBEAR. Slightly annoyingly, for the purposes of demonstrating, by default, if I click on the link that is provided, it will open this connection in a new window, which I don't want it to do. So I'm going to right click on it and say connect to hosting current window. And then all being well, press my password. and connect me through. So I can now navigate around my home directory, which is where it by default will place me. But as you can see, I can edit that entirely. I have symlinks to my project directories from my home directory for various different tools. So that's generally kind of advised practice, particularly if you're using anything like Jupyter Lab on their portal, where that's a requirement if you need to get around. But for now, I just have some basic templates in here. So I can open that directory. It will ask me, I believe, for my password again at this point. So if you don't have SSH keys set up for connecting to BlueBEAR, my advice would be to do that because it speeds up this back and forth a little bit. And then eventually it will drop me onto that directory, and then I can edit the files as required using the native version of VS Code running on my computer. So it's a more feature-rich editor than maybe otherwise available. You can install the extensions. So the extensions are broadly compiled for x86. If you're trying to VS Code onto one of the Power 9 nodes, that may be more problematic because not all of the binaries will be compiled for PowerPC 64LE, I imagine. I think more of them are being compiled for ARM systems. Sort of watch the space for that, but at the moment, certainly with BlueBEAR, you're fine because the majority of it is x86. Like I say, you can put all the extensions that are available. The majority of extensions that are available will then get installed into essentially the remote VS Code server that is now running on BlueBEAR. Now, I mentioned earlier the caveat here, and the same applies to Tmux as applies to VS Code, is that this is running on one of the login nodes. So there are resource limitations on the login nodes. Just be aware of what you're running. If you're running extremely kind of intensive computational extensions, then you may get an automated message and you might need to shift the workflow around a little bit. I am looking at the moment at how possible it is to integrate this VS Code SSH with our sort of backend Slurm system, and there is some work being done in the community around this as well, because that would be a useful feature, I think. But the ability to connect from a local running VS Code, but actually working on the server, executing your code on the server, is a handy thing to do. So that's running VS Code natively. I mentioned there is another option, which is to essentially run it via BEAR portal. I'll shift over to this screen. I have my BEAR portal instance, interactive apps, it's a place to go, and then code server. So code server is a sort of server client. I think I missed that click. Let's go this way. I got it. I'm just being impatient. And now I've overridden it. There we go. So sorry, it's a client server essential version of VS Code. I will start it with the default settings, so just an hour for now. So the big advantage to this is that it will launch me a VS Code server and connect my web client to it, but it is running on a BlueBEAR compute node, not on a login node. So you don't have the resource limitations that you do, and you won't get automated messages to ask you not to do it. And it should launch fairly quickly. And as you can see, it looks very similar. So it's put me again into my home directory. I can install extensions in the same way. I don't have access to all the same extensions. The way that the code server works is slightly different, but they're constantly improving that. But it looks a very sort of similar environment to running. Obviously, it's running natively in your web browser, so it's not forwarding the graphical connection. So it's not forwarding the VNC, which is what some of the other apps such as MATLAB, how they work. So it's generally a more responsive experience. So that is Tmux, that is VS Code. Does anyone have any questions regarding VS Code running? We will likely put some documentation around this to kind of to advise on good practice around using it. But if nobody has any questions for now, I will just end with the final slide, which is please complete our survey. And hopefully I've made that QR code large enough so that you can actually use it from within the room. There we go.
Thank you. Thank you to Jess. Thank you. Excellent. Thank you all. As always, we're trying to create this community to try and help ourselves and everyone else so that we can create this community. If you guys have any kind of talks you want to give or any kind of subject matter you want to kind of discuss, please do let us know. We'll be happy to try and find people, or even if you can just give us a brief talk about it, we'll be happy to put that in. We have this room booked for another hour, so if you still want to stay around and talk to other people or grab lunch, feel free. As I said, we'll put the video up and slides on our Canvas pages. The next one is our basketball talk, which will be very interesting regarding the work that the guys are doing, especially around the GPU work, and just to demystify the GPU stuff we've been putting in, and also the cookies and coding session in July. We then have a break in August. So we're looking for talks in September, October, November. So please feel free. Thank you all.